and we will talk about brain photobiomodulation. So a very interesting emerging method to keep our brains young, healthy, and sharp. I think most of you have heard the term photobiomodulation. It's an umbrella term for medical light therapy. Huh? So many people talk about photobiomodulation as low-level light therapy or low-level laser therapy, maybe even cold laser therapy, soft laser therapy. But the established umbrella term is now photobiomodulation, PBM is the abbreviation. And yeah, we will talk specifically how we can apply it to our brain and how it can support our brain health. So don't worry if you never heard about PBM. I will give you fundamental knowledge, give a little bit of context, also about the key aspects of a healthy brain. And then I will dive deeper into the mechanisms of brain PBM. And yeah, I will show you the most important studies to give you an overview about the current state of research and the areas of application. And yeah, overall, I hope that my lecture will give you a good understanding of what brain PBM can do and if it could be an interesting tool to be used in your clinical practice. So first of all, why am I qualified to talk about all that? So first of all, I've been working in the field of photobiomodulation for 13 years now. I'm part of Weber Medical, a German-based company. We produce laser technology. I'm also a board member of ISLA, the International Society for Medical Laser Applications. I'm a co-author of 3 editions of a book called Medical Laser Therapy. Also a co-author of a study on photodynamic therapy. And yeah, I hold a postgraduate diploma in neuroscience and psychology from King's College London. So yeah, brain PBM really is my favorite area of the whole field of photobiomodulation. But before we go into it, let me tell you about the foundations, the fundamental knowledge you should have about PBM. So first of all, in order for light to have an influence on our human biology, we need light absorbing components in our human body. We need photon receptors, and these are scientifically called chromophores. So we have different chromophores all over our body. And what is important to know, different colors of light are absorbed by different chromophores. And that is why different colors of light have different effects on our human body, very generally speaking. On the right side, you see a rainbow. And what I would like you to take away, the colors you see at the bottom of the rainbow, UV light, blue light especially, then comes green, yellow. These are the colors with limited penetration depths into human tissue. So most people talk about red light and infrared light therapy. That has become a very common term. And few people talk about UV light, blue light, green light therapy, just because it's not so common and you have to use those colors of light normally in an invasive way. Now, of course, you can use them to treat certain skin conditions, but if you want to bring them into the depth of the human body, it has to be done in a minimal invasive way. An important term I want to mention is the term wavelengths. Wavelengths simply describes the color of light in nanometers. So just to give you an example, we will focus on infrared light today, and that starts in a range of approximately 800 nanometers. And below the rainbow, you see a little overview about the wavelength ranges of different colors. So we will focus on infrared light therapy, and here is information about the chromophore for red and infrared light. It's an enzyme within our mitochondria, specifically in complex 4, and it's called cytochrome C oxidase. I think, of course, all of you know about the basics of what is happening in our mitochondria. The energy we eat in form of electrons and the oxygen we breathe all that comes together and is transformed in our mitochondria to ATP. ATP is the only energy currency that our cells can use. And what happens specifically in cytochrome C oxidase, here oxygen is reduced to water. And so we need a lot of electrons for that reduction process. And when that happens, a lot of energy is released, and that energy is used to pump protons out of the inner mitochondrial membrane to the outside. So that will establish a gradient, a difference in voltage between the inside, the inner part of the inner mitochondrial membrane, and the outside. And whenever in nature we have a gradient, there is a natural tendency to balance that gradient out. So photons want to move back into the inner mitochondrial membrane. And the only way to do that is to go through what is called ATP synthase. So here, ATP synthase acts like a turbine. It rotates when the protons move back in. And yeah, that rotation releases a lot of energy, and it's a pretty complicated process. I'm not going to explain all details, but with that energy in that process, ADP is converted to ATP, the energy currency we need on a cellular level. So yeah, these are the chromophores for red and infrared light. And another fundamental thing about photobiomodulation, you can use both lasers and LEDs as light sources. But generally speaking, to keep it simple, lasers are a bit superior because the light beams comes out as a very focused, coherent light beam. So it has better penetration abilities into human tissue, while LED light scatters. Yeah, it kind of diffuses in all different directions, and that will limit the penetration depth a little bit. Here's an overview about how you can apply PBM. There are non-invasive applications. We talked about brain photobiomodulation. It's also very typically used to treat larger pain areas. So here people would use patches or sometimes go into light beds to have a full body irradiation. Laser acupuncture is also a very common thing. And of course, the whole field of dermatology. A lot of lasers are used in dermatology. And if you really want to use the shorter wavelengths, you can use intravenous, intra-articular in joints, or interstitial applications. So basically, we nowadays have technology to bring the light into really all parts of our human body. And the system you would use is called the endolaser, or now combi laser system from Weber Medical. There's a very specific kind of photobiomodulation called photodynamic therapy. So with the previously mentioned applications, we always want to stimulate or activate or regenerate tissue. But in photodynamic therapy, we have a specific target that we want to destroy. So it could be bacteria, viruses, fungi, parasites, or cancer cells. But I will not go into more details today. It's not our topic, just to give you a little bit of an overview. And here you see how brain photobiomodulation is normally applied. So we use helmet-like products mostly. And as I mentioned before, we have to use infrared light. It's the only light that can really penetrate through the skull, and it will be absorbed in cortical brain structures. So, of course, a lot of the light energy will be absorbed on the way. And to give you a ballpark number, approximately 2 to 3% of the energy you applied on the surface will reach a depth of 2 centimeters. So it depends on the brain area. In some areas of our brain, we have higher percentages, could be up to 10 or 12%. But mostly it's around that. So yeah, this is just for the back of your mind. You have to calculate first how much energy you apply to the skull, but then you also should know how much of that energy reaches the desired brain area. So I told you already, it's a mitochondrial therapy. And what I will tell you today is that it can treat many of the fundamental causes of brain aging or of brain disorders. So it's yeah, very commonly used now as a longevity hack, brain longevity, also used in integrative neurology. And yeah, you will also see that it's very easy to use and safe to use even at home by patients. Okay, that was a little bit of context about photobiomodulation. I want also to give you context about yeah, what makes up for a healthy versus an unhealthy brain. So I will be very quick with that slide. Of course, there are many factors that yeah, influence how healthy our brain is. Lifestyle factors, sleep is so important, movement, exercise, our diet, our stress levels. Yeah, but I don't want to lose too much time here. You see just that overview, and what is interesting is the manifestation on the level of the brain. So yeah, I chose to differentiate four categories here. We have the category of energy, and that, of course, is about very much about mitochondrial function. But it's also about how well we can supply nutrients and oxygen to our brain. Let me go to the next slide, because here you see already what we will talk about today. So what you see in green will be a focus of my lecture, and what you see in yellowish will be at least mentioned in my lecture. So we have the category of communication, and yeah, I will briefly mention intra and intercellular communication, also how, yeah, of course, how that is influenced by PBM, also how epigenetic regulation is influenced by PBM. PBM can also influence our brain waves and our brain network communication. There are, of course, in general, more aspects to be considered. Third category, structure. Yeah, we will and state of our brain. So we will really focus on inflammation, on neuroinflammations, and yeah, I will dive a bit deeper on that. And in my next slide, we will also mention oxidative stress, our vascular architecture and integrity. And yeah, related to the topic of neuroinflammation, yeah, I will tell you about our glymphatic system, our waste removal system in our brain, and how PBM can enhance our glymphatic system. And we will also tackle how yeah, PBM can yeah, activate stem cells, how it can enhance neuroplasticity, and it can also has an influence on our proteostasis. So that means on the proteins we have hopefully in yeah, sufficient abundance in our brain. Okay, let's dive a bit deeper into neuroinflammation and why it's a key player in neurodegeneration. So really, neuroinflammation really has emerged as yeah, a common denominator of not only all neurodegenerative disorders, but basically all brain disorders. And yeah, the old thinking was kind of okay, there is neurodegeneration, there are protein accumulations like the famous beta amyloid protein accumulations in Alzheimer's, and that will cause inflammation. And the new thinking is now shifting a little bit. And there are now many studies suggesting that inflammation might come first and yeah, might be a causing factor for protein accumulations, but also other neurodegenerative processes. So, of course, in general, an inflammatory response is healthy and very important. It's triggered by our immune system. When our immune system recognizes DAMPs, these are damage associated molecular patterns, yeah, could be caused by toxins, brain injuries, by cellular stress, for example, or PAMs, pathogen-associated molecular patterns. And yeah, these would, of course, as the name suggests, be caused by viruses, bacteria, fungi, parasites. And so that will trigger the inflammatory response, will lead to the elimination of danger if things go well, and the inflammatory process, finishes. But sometimes our immune system fails to resolve the cause of the inflammation or is activated again and again. So why would it be activated again and again? For example, because of chronic exposure to toxins, or because you have systemic inflammation, or because you have a brain injury, or could be caused by gut problems. Yeah, the gut-brain barrier is very important, and that will lead to a vicious cycle. So we have continuous activation of immune cells, of glia cells and astrocytes, and that will lead to a shift in the microglia phenotype. Yeah? So we will have a higher prevalence of a pro-inflammatory microglia phenotype. That phenotype has an elevated baseline inflammatory state, and it will react quicker and more strongly to inflammatory responses. Even if there's only a small and maybe not even, normally not even significant stimulation. So we have that vicious cycle that kind of reinforces neuroinflammation and prevents our brain from going back to normal, going back to homeostasis. And the consequences of chronic neuroinflammation, you see that highlighted here, they include impaired blood flow and perfusion to the brain, poor oxygenation, mitochondrial dysfunction, abnormal synaptic pruning, oxidative stress, excitotoxicity, impaired protein clearance, and connected to that, more protein aggregation, disrupted blood-brain barrier. And yeah, that's not even the entire list, but in the end, it will trigger so many processes that will, lead to neurodegeneration. Here's one example how that specifically works in Alzheimer's disease. So in that image, the beta amyloid aggregations, hope you can see my mouse cursor here. So they come first here in that image. If that's really the case, is an open question right now. As I mentioned, inflammation might come first. But anyway, what you see here, the activated microglia, they can potentially clear the beta amyloid accumulations, and they come to rest again. Our brain goes back to homeostasis. Or they can fail to clear the beta amyloid accumulations. And we see what we just talked about. We see chronically activated microglia, and they will emit and release different molecules that will further stimulate inflammatory processes. For example, APP. APP is the amyloid precursor protein. Yeah. So here we have the first feedback loop, and there's a second feedback loop. Chronically activated microglia, they will, for example, release more pro-inflammatory cytokines like interleukin-6, for example. If we have those in excess, we see increased neuronal death. And dying neurons, they will then again emit different materials that can are recognized as DAMs and will trigger the inflammatory response. So takeaway: neuroinflammation is a very important hallmark of brain disorders. And we will see later that it can be treated by brain photobiomodulation. Another thing I really want to highlight a little bit is how important mitochondrial function is. So, of course, ATP production is super important, especially our brain with a very high energy consumption relies on, uninterrupted ATP supply. Yeah, even a very quick temporary ATP shortage can have devastating consequences in our brain. But there are many more functions of mitochondria in our brain specifically. So we kind of used to look at mitochondria as only the powerhouses of cells, but yeah, that notion is really outdated. And now we understand many more functions. For example, they have to balance reactive oxygen species. Of course, we need oxygen in complex 4 of the ATP production chain. But sometimes the oxygen molecules react prematurely along the respiratory chain. Yeah, that mostly happens in complex 1 and 3. In 2, it's not very common. And if it happens, yeah, it leads to the development of reactive oxygen species. And what is important to know, mitochondria do have a built-in antioxidant system, but it can run out of balance. Yeah, mitochondrial dysfunction often leads to that system coming out of balance, and that will cause oxidative stress, oxidative damage. Another thing that is very important, mitochondria have their own DNA. So with that DNA, they produce specific proteins that are yeah, can only be produced within our mitochondria and that are very important for brain function. Mitochondria, they divide by fission, meaning one becomes two. And they also fuse from time to time, so meaning two become one. And that will, of course, has yeah, a lot of membrane dynamics, and that these membrane dynamics will have influences on other organelles. So another thing that could go wrong potentially. And we already talked about neuroinflammation. Our mitochondria contain a lot of potentially pro-inflammatory material. If too much mitochondrial RNA or DNA or reactive oxygen species, as I mentioned, leak out, our immune system will recognize them as DAMs or PAMs, and so mitochondrial dysfunction can be a cause of neuroinflammation. And here you see a slide about a new model. So mitochondria are also modeled as information processing units. Yeah, we call it the mitochondrial information processing system. So in simple terms, it describes that mitochondria kind of sense the outer environment of our cells. Yeah, they look for metabolic, biochemical, and neuroendocrine signals, and they will convert those signals through metabolites and signaling molecules into signals that can be read and understood by other organelles or also by our nucleus, where we have, of course, 99% of our DNA. So mitochondria tell our nucleus about the outer environment of our cells, and that of course has a big influence on our gene expression. So in other words, mitochondria also act as very important epigenetic regulators. Yeah, and really takeaway, I'm repeating myself now, the image, the analogy of mitochondria as powerhouses is not up to date anymore. Yeah, and here you see a slide just showing you that mitochondrial dysfunction really is related to all kinds of brain disorders, not only degenerative disorders, also mental disorders, brain injuries, the normal process of aging, and even brain cancer. Yeah, another hallmark I want to highlight is how important cerebral perfusion is. And here is a population study on almost 5,000 participants. And the takeaway is simple. Lower cerebral perfusion is associated with a higher risk of developing dementia. And here there is another study just showing mechanisms behind that finding. So these mechanisms would include that impaired blood flow to the brain can lead and can induce to oxidative stress, more protein accumulations, synaptic dysfunction, neuronal loss, white matter lesions, and neuroinflammation. Okay, I created a little bit of context now, and based on that context, I would now like to dive deeper into brain biomodulation and focus on the details of what happens when photons are absorbed in cytochrome C oxidase. So maybe interesting for you to know, it's specifically the copper ions in CCO that are in the end our chromophores. And what happens when photons are absorbed are two things. First of all, electrons are being released, and we talked about it before. They are very important for the reduction process from water to oxygen. So having more electrons means that we can more efficiently, more quickly, and run those reduction processes. Another very important aspect is often we see nitric oxide, NO, bound to a cytochrome C oxidase. And when photons are being absorbed, we observe what is called a photodissociation of nitric oxide from CCO. So in simple terms, NO is released from CCO, and so NO would be a potential blockage in the ATP production process. And by releasing NO, we remove that potential blockage. And these two mechanisms explain why photon absorption, light absorption by CCO can directly lead to increased ATP production. I will show you a study later, but there are different studies on different cell cultures, and to give you a number, depending on the type of cells, ATP production can be increased by approximately 30 to 100%. So pretty significant numbers. There is a second primary direct effect because our brain, our mitochondria, can now metabolize more oxygen. It will signal back, to put it simple, to our body that more oxygen is now needed in our brain, so we can improve oxygenation in our brain. And that is our second primary effect, and there is also a third one. I mentioned that nitric oxide is released from CCO. And you probably know NO has many different signaling functions in our human body, but maybe the most important is that it signals for vasodilation, for a widening of blood vessels, and that's a mechanism of why we see increased blood flow to our brain after brain PBM. So here are a couple of studies just demonstrating what I just explained. These are examples. What you look at in studies, if you want to demonstrate the effect of that I just explained, you look for oxidized cytochrome C oxidase because they lost electrons. So that was demonstrated in many studies, for example, in this one here. And what was also observed is what I also mentioned: improved cerebral blood flow, blood oxygenation, and of course, by that also improved blood volume. Here is that study I already mentioned. So they used a light of 808 nanometers. They shown it on human neural progenitor cells. So it was a cell culture study, and what they observed, the output of ATP units almost doubled in the in those cells that were irradiated by light. You can see the numbers here in the laser-treated cell group. The quantity of ATP units was around 7,500, which is almost double the amount of the non-irradiated ATP units, ATP cells. Okay, here is a review, showing you that there's a lot of animal and human studies in that field, and summary of that review was that brain PBM is recommended to increase cerebral blood flow and metabolism and oxygenation. And also, that's for now just a side note, it can also be used to modulate brain waves, brain neural oscillations. But I will come back to that in a minute. For now, I would like to focus on what we talked about, neuroinflammation, how brain PBM can reduce neuroinflammation by stimulating our glymphatic system. So before I explain you the mechanisms, here is an animal review. 27 animal studies were reviewed, and what you see here, simple takeaway, PBM could upregulate anti-inflammatory cytokines and downregulate pro-inflammatory cytokines. And it could also change the phenotype of microglia. We saw more anti-inflammatory microglia than those with the pro-inflammatory phenotype. So here you see an image of the glymphatic system. Maybe first of all, why is it called glymphatic system, not lymphatic system? That G is added as a reference to glia cells, our main immune cells in our brain. And also the whole system works a little bit differently than the glymphatic system in the rest of our body. So how it works in a nutshell, there is a cerebrospinal fluid, which is actually 99% water. And first it flows in the perivascular space next to the arteries, and then it flushes into the interstitial space. You see the green arrows here. And in the interstitial space, it picks up debris, garbage, toxins, waste products. And then it moves back into the perivascular space next to the veins. It will be led to the actual typical lymphatic vessels that we start having in our meninges, in the outer membrane of our brain. And what is important, when the cerebrospinal fluid flushes into the interstitial space, it has to pass through water channels called aquaporin 4. And one of the mechanisms why brain PBM can enhance glymphatic system function is because it can make those aquaporin 4 water channels more permeable. That is one very interesting mechanism. There are 3 more mechanisms. So a part of infrared light is also absorbed by water, and it changes the structure of water molecules. And I mentioned already the cerebrospinal fluid is approximately 99% water. So it flows more freely after brain PBM. Another mechanism is related to what we talked about, to nitric oxide. The lymphatic vessels in our meninges, they are also dilated after PBM and can more effectively transport the cerebrospinal fluid. Plus, there's a fourth mechanism. So when more blood flushes to our brain, of course, it does not increase the total space we have in our brain. So there's now more blood, but the same amount of space. And that creates a pressure that pushes the cerebrospinal fluid out of our brain. Simply because more blood needs more space. So that's a very easy to understand, very intuitive mechanism. So everything we talked about, let me summarize. Increased ATP production, enhanced oxygenation, enhanced blood flow to our brain, and reduced neuroinflammation. Yeah, these four aspects are super fundamental to brain health. We kind of change our whole brain environment, our whole milieu, and yeah, make it a healthier milieu. And of course, that will have a cascade of downstream effects behind it. And these downstream effects, they include increased neuroplasticity. We specifically observed an increase, a upregulation of BDNF, the brain-derived neurotrophic factor, and that is directly correlated to enhanced neurogenesis and synaptogenesis. Also, what is a pretty well-known and commonly discussed aspect of photobiomodulation, we can activate stem cells. And of course, that is a very important mechanism for neuro regeneration. We mentioned already that we can modulate the epigenetic regulation because of the function of our mitochondria, but also because the whole brain environment has changed, is now more healthy. So we influence our genes in a beneficial way, our genetic expression. And there's also an antioxidant effect, and we can alter network dynamics. And I want to talk about two of those aspects in more detail. Here, this is about the gene expression. This is a rat study, and they irradiated the area around the spinal cord of those rats. And what they could observe is that 1,275 genes reacted to PBM. Some were upregulated, and those include very important longevity genes. You can see them here listed. And other genes were downregulated, and these were mainly genes correlated to apoptosis, for example. In conclusion was the way PBM influenced our gene expression, or the rat's gene expression, was in a very favorable way. And here is one slide that is about brainwave entrainment. So, meaning how light changes our neural oscillation, our brainwaves. And what is very interesting here, you see they used wavelengths of 1064 nanometers on continuous wave. So continuous wave means that the light beam is not interrupted. There is no frequency on that light beam. So this is a standard. This is what you normally use. And even that non-pulsed, no frequency light beam had an influence on alpha and beta waves. Yeah? And what is, in my opinion, very interesting, when you put a specific frequency on the light beam, you can entrain brainwaves. There are resonance effects. And for example, putting a frequency of 40 hertz will stimulate gamma waves because it's within the gamma wave range. So we have those very interesting resonance effects. And I think the future of brain PBM will for sure be to couple it with EEG and to observe which brainwaves might be underrepresented in certain brain areas. So to specifically stimulate those brainwaves. So I think really in a couple of years, maybe a decade, it will go down the path and become precision medicine. Okay, I hope I could explain how brain PBM works. And now I would like to move on and show you the most important studies. And the first domain I would like to talk about is dementia, and of course including Alzheimer's. And here you see a review that was conducted on 36 studies, and 17 of those were on animals, 9 on cell cultures, and 10 on humans. And what is pretty remarkable, all those studies reported beneficial results. So in humans, they would, of course, first in line include improved cognition. What was also highlighted in that review, there was a lack of any adverse effects and a remarkable ease of use. So remember, this is about dementia patients. So really patients that might have a hard time in daily life to function in daily life. And the devices people can use are so easy to use that even dementia, Alzheimer's patients can use that therapy at home. Yeah, what has to be said also that most studies had only small patient numbers and not all had highest quality. So there's still more research to do, to call it like final evidence. But here in that field, we really have very good preliminary evidence, but also a call for larger trials. But in addition to the good preliminary evidence, we have a very sound mechanistic rationale of why it should help dementia patients. Here's one study, just to show you some details. A study conducted on 57 patients. They received brain PBM twice a day, each time for 6 minutes, daily for 8 weeks. And you can see here what happened to those patients after already 7 days. Many reported improved sleep quality. And after 2 to 3 weeks, most of the patients had improved mood, improved cognition, higher energy levels, less anxiety. So, pretty quick results also. And what you see here on the left side is a little demonstration, like patients were asked to draw a clock. On the left side, you see what they were able to do before treatment. And then the same patients were able to draw much more detail and much better drawings of clocks after treatment. So I find that a nice illustration. Okay, here is just an overview about how BrainBM should work on dementia and Alzheimer's, but I think we talked about it. I will move on. And yes, now we talk about Parkinson's disease. Okay, I wanted to show you a video, but I think I will skip it because I'm running a little bit late. So what you would have seen are interviews of patients that were part of this study you see here. It was a pretty small study on 12 patients only, but it was a very high quality study. And what you see in bold are the outcomes after 3 months of brain photobiomodulation. Sorry, not only brain photobiomodulation, they also used intranasal, meaning systemic PBM, also neck PBM and abdominal PBM. So why abdominal? Because the gut microbiome plays an important role in the development and onset of Parkinson's disease. And the neck, because from there, we can more easily reach the basal ganglia, which is the affected brain area, and it's pretty deep in our brain. So we will probably not reach it by just transcranial application. They also added neck application, and so now I'm started talking about it. So when we talk about Parkinson's disease, we always wonder a little bit, okay, why do we see such good results on Parkinson's, even though the light applied to the skull will probably not reach the basal ganglia? And the explanation is that we can influence brain networks, especially here by treating the prefrontal cortex. We can influence brain networks that go down to the basal ganglia. and have more or less a systemic effect on the brain. Okay, that might be a bit too detailed right now. I'm working on an online course that about brain photobiomodulation. It should be ready in May or June, and it will go yeah, into those details. And yeah, maybe you will be interested in it. Anyway, what you see here, the results include yeah, improved mobility, improved cognition, um, better dynamic balance, and better fine motor skills. And what was also checked here in that studies, that those improvements could be maintained as long as the treatment continued. And later, the same research group conducted a follow-up study of that initial study. And here, 8 of the initial 12 patients yeah, took part in that study. And these patients had continued PBM treatment at home 3 times a week with a home use device. And what you see here is that the results not only maintained, but that they observed further significant improvements in walking speed, in stride length, in different tests, and yeah, in general in cognition and dynamic balance. So this is a very encouraging, very interesting result, especially if you keep in mind that, yeah, once you have Parkinson's, it normally only becomes worse, and you hardly don't see any improvements. So a very interesting study on, yeah, very few patients only, but I think that Parkinson's will be a very common indication for brain photobiomodulation. And yeah, since those initial studies, many more studies were started or even published by now. You see here one that was published last year on 36 patients. And also here, conclusion was, yeah, they observed improvements in mobility and yeah, motor experiences in daily life and a reduction of anxiety. Okay, we talked about treating neurodegenerative disorders so far, but of course, PBM is also an ideal intervention to prevent getting or developing those neurodegenerative disorders in the first place. And yeah, that is highlighted by the fact that we could observe improvements of cognitive function also in healthy individuals, yeah? Not only in people with brain disorders, also in healthy individuals. Here is a review on 35 human studies. And yeah, parts of them were conducted on dementia patients, some on patients suffering from brain injuries, but as I mentioned, some also on healthy people. 29 out of those 35 studies reported beneficial results that yeah, included enhanced attention span, improved reaction time, and improved memory performance. So this is also yeah, an interesting field of application. Here is another one, brain injuries. Here is also a review. That review was conducted only on human studies. And yeah, you see an overview about the results that were summarized. So brain PBM in traumatic brain injury can lead to improved cerebral blood flow. It can improve cognition once again, and it can improve functional outcomes after mild traumatic brain injury. And also what was also reported reduced markers associated with apoptosis and immune cell activation. Okay, I have 2 more domains I would like to briefly talk about. So the 5th one is mental disorders. Here we have a review on brain PBM to treat depression. 11 human trials were summarized, and so everything we talked about so far, we had across the board good results. It's a bit more mixed for depression. So here we had some studies that did not report positive results, but most likely because the applied energy was too low. Yeah, so here we could establish a minimal energy threshold I'm not going into details, but we need a certain amount of energy. I mentioned that before. Otherwise, nothing's going to happen. And yeah, that summary concluded that we have moderate evidence that PBM is effective for depression. And yeah, there are more trials on the way. But especially if you consider that research is now starting to look at depression also as a metabolic disorder. Yeah, it makes a lot of sense that brain PBM can be useful. But of course, depression can have so many different causes and details, nuances to it. So I think, yeah, it can be useful and you should combine it. And yeah, that is also what was also concluded in that review, because they not only looked at brain photobiomodulation, they also looked how systemic intravenous photobiomodulation could influence symptoms of depression, and concluded that systemic intravenous PBM was even superior to the effects of brain photobiomodulation. T means here transcranial photobiomodulation. So ideally, you can combine both applications. And here below, you see Dr. Weber giving an IV laser therapy to a patient. Here's a study on anxiety, and I will hurry up a little bit. You can see in bold the conclusion, significant positive effects on anxiety measured with the Hamilton Anxiety Scale after 2 months of brain PBM. And yeah, PTSD might emerge as another field. Here you see a small study on traumatized firefighters, and they were treated for 8 weeks. And yeah, in the end, um, they concluded that was a good proof of concept study, and yeah, no side effects, good compliance, and yeah, the life quality and situation of those firefighters could be improved. Last domain I would like to talk about is autism spectrum disorder. I chose 2 studies as examples. So the first one was conducted on adult people between 18 and 59 years old. They received twice a week brain PBM for 8 weeks. And yeah, what was reported were improvements in social awareness, communication, motivation, and less restricted and less repetitive behavior. And what is most important, in my opinion, quality of life of those patients improved. And here you see another autism study, and here this one was conducted on young people between 5 and 15 years of age, with an average of 9.1 years. And yeah, after 6 months of treatment, it led to a significant reduction of autism spectrum disorder severity, including a reduction of non-compliant behavior. And also interesting to note, the stress of caregivers, of the parents of the family, was reduced. Okay, you see, we have really different fields of application for brain photobiomodulation. And I think that makes sense because it works on a very fundamental level, on very fundamental processes that make up for a healthy brain. And yeah, that's why we can use it for really a number of different applications. So last thing I want to show you today is the product we at Weber Medical just launched. So it's an infrared helmet. We call it the Weber Brain. And very important here with the Weber Brain, we only use real laser technology, no LEDs. Yeah, and as I explained before, that is very important because a laser light has much better penetration properties through the skull. So more of the energy that we apply on the skull will actually be absorbed in cortical brain structures. So that is a big improvement, and related to that, there's now a very sophisticated lens system built in, And that lens system focuses the light even more, so that further enhances the penetration. There's also what we call a light finger technology. So what does it mean? It means that there are little fingers, little protrusions that we can, with which we can bypass the hair. So we can put those fingers directly on the skull. And that is also important because hair can absorb a lot of the light energy. If we're talking about dark hair, it could be up to 80%. So it's very important to use a tool that can bypass hair as much as possible. And one, maybe the most important aspect, we now use three wavelengths simultaneously. 680 nanometers is red light, so it will not really reach cortical brain structures, but it can help to improve blood flow to the brain. And it's also for another purpose because it can very effectively stimulate hair growth. And then we use the two typical infrared wavelengths, 808 nanometers and 1064 nanometers. So if you dive deeper into the research or maybe listen to my online course at one point, you will see that the best results were achieved with those two wavelengths. So actually it was a no-brainer. It was an easy choice to use those wavelengths. The Weber Brain is available in two different versions. One is a home use version. So everything I talked about applies to both versions. What is different? The professional version has 3 times more output power because it has approximately 3 times more diodes built in. Plus, it allows you to put different settings on different brain areas. Yeah, you can, for example, you want to put a frequency of 10 hertz and use it to treat the prefrontal cortex, but maybe you want to use 40 hertz on the occipital lobe, just as examples. So the Weber Brain allows you to do that in a very sophisticated way. Yeah, you can run it by an app, and it really allows for precision medicine. And I think I mentioned before, the whole field is certainly moving that way. The home use is mostly an easy to use device. So it's designed for patients to use it at home. So you just put it on your head, you turn it on, it will irradiate the whole head, and after 15 to 20 minutes, therapy is done. Not much you can do wrong. These are the differences. Of course, there's a difference in price. And with that, I'm coming to the end of my lecture. So I want to leave you my contacts and how you can find more information. So if you really want to dive deeper into the field of medical laser therapy, we have now launched the third edition of our book. So it covers all different areas of application, from non-invasive to invasive photobiomodulation to PDT, but is also maybe first in line a very good practical guideline. It has a very comprehensive protocol section. So if you want to use our technology, I recommend you buy that book as well. And as I mentioned before, I will soon launch my online course that will go into much more detail about brain photobiomodulation. Okay, this is how you can contact me. Best ways are by email or maybe also connect on LinkedIn. These are my two favorite ways of connecting to you guys. And thank you for your attention.